Good afternoon, girls. We are going to start our class that we are going to solve May, June 17, variant 43 that I have given you this. Yesterday I gave you the link. So before starting my class, I want to request you all to clear your dues because till March, if you are not clearing your school fees, after 10th, you are not allowed to enter any virtual classes. So girls, keep this thing in your mind, clear your school tuition fees. Now, I'll share you the screen and everyone look at the question. So this is our past paper. Yes. No, that actually for everyone the same question, Anita, that's why you are not withdrawing. Everyone is actually, do I feel that if you want to give the exam up to number, you can do the registration again, no problem. But ask yourself, ask your parent what will be good for you. Take the decision according to that. Actually, nowadays we are in the situation that what will be good, that also you couldn't understand. The way people are dying, if you see the death rate, every day, thousand people are dying. So in this critical condition, taking decision is very tough. Okay, if anyone wants to talk, you can unmute your microphone and one or two can talk. But because if you are talking everyone together, then it will be a mess. So this question, look at question number 1. This figure 1.1, this is a diagram. Girls, this is the diagram. Shows the phase during the heartbeat when the atria contract, the blood vessels. Someone is writing.
It's Anika.
Yeah, I understood.
Hey, Anika, stop writing messages.
Okay, so if you have anything, I will discuss, don't worry, but now let's start our class. So girls, this is the diagram. If you are noticing the leveling, they're asking the arrow to show the pathway taken by the blood from a vein into the right ventricle. So this is the right ventricle. So you can draw the arrow from A and then show it the right ventricle. Or you can draw the arrow from B and show the right ventricle. Because thus, a right ventricle from the superior vena cava or inferior vena cava, from both the part, the blood can come and enter to the right ventricle. So both the path they are showing, so you can do it. Now, next question. Identify the letter of the blood vessels that carries blood at the highest pressure and with its name. Look, highest pressure means that it will be aorta, right, girls?
Yes.
So if you are looking at this diagram, tell me which blood vessel shows aorta?
C.
C, yes, C for cat, C, yes. So here they're asking, suggest what causes the valve 1 and 2 close during the heartbeat. Girls, valve 1 and 2 is not atrioventricular valve.
Valve 1 and 2,
semilunar valve.
Semilunar valve, when it will be closed? When the ventricle relaxes, right?
Yes.
When the ventricle relaxes and it increases the volume of ventricle, because relaxing time, it will the volume will increase, right? So
yes,
the pressure will be higher in aorta and pulmonary artery, right? So higher pressure will be in the aorta and pulmonary artery, then the ventricle, and this causes semilunar valve to close. So I explained, see, that when ventricles are relaxing, they are increasing their volume. So the pressure will be higher in aorta and pulmonary artery, because from pulmonary artery, blood will go to the lung, and through the aorta, it will go to the body cell, right? So that when the pressure will be more in the aorta and pulmonary that artery compared to the ventricles, this causes semilunar valve to closes to prevent the backflow, right? So the blood will not come back to the ventricle again. So this is the question, state the function of the valve 1 and 2. This valve is everywhere, they're doing the same function, stop backflow of the blood, right? Now look at this question. Figure 1.1 shows the phase of the heartbeat when the ventricles are filling with blood. Describe and explain. Girls, 2 points. And total mark of this question is 4. Describe and explain how the blood travels from the right ventricle to the lungs. We'll go back to the diagram. See, right ventricle. So, right ventricle. Girls, most probably next week I'll take a written test. I'll give you the
test?
Yeah, written test. I will give you the syllabus and all that, or multiple choice or whatever the question. Let me think in this weekend, but get ready for the test. Don't say that Miss because
Will be able to?
I think it will be mixed. Okay.
Okay.
So get ready for the test. And now the question they're asking, describe and explain how the blood travels from the right ventricle to the lung. Look at the arrow. So right ventricle here, and through the pulmonary artery you go to the lung, right? So but this that pathway you have to explain with the letter or with the part of the lung by using the letter, by using the name. Let me explain this, and you people please look at the diagram and follow the arrow. When the right ventricle contracts, it increases blood pressure, right?
Yes.
So the blood pressure is higher in the ventricle than the arteries. So this causes the semilunar valve or valve 1. As we are working on this side, right side of the heart, we will not talk about the valve 2 because our question is not that. So it causes semilunar valve or valve 1 to open. Then the blood flows from the right ventricle to the pulmonary artery or D/E because these are the 2 branches. Okay, understood. Now this is that look at this, that it causes that semilunar valve or valve 1 to open, then blood flows from right ventricle, okay, to pulmonary artery or D/E. Semilunar valve closes. This semilunar valve will closes when the blood in pulmonary artery, because ventricle relax and stop blood flow in the blood. Through the pulmonary artery, blood moves to the lung. Clear?
Yes,
Yes.
Now, this actually, if the circulation, that if you can understand circulation, it will be easy. It's not a problem. Now, someone drew arrow. Could you please remove this? It will come to our every page. The one drew, please remove this. Remove this arrow also. Now, next question. State the name of the part of the heart that level 3. And they are asking your name of that part and the role. Let's see the diagram. Look, tell me the name of 3.
Oh, it's the septum.
Excellent.
Septum.
And what is its function? Role?
It separates
separates the
oxygenated
blood.
and deoxygenated blood.
Okay.
And it also allows double circulation. Excellent. Thank you. Your answer is correct. Now, we will be moving on next question, question number 2. And in this question, see, quinoa. This is Synopodium quinoa. This is the scientific name of quinoa. This is a one dicotyledonous plant that produce seeds that resemble those cereal, like rice. This is cereal type 1 grain. People are using alternative of the rice, but we people love to take rice only. Now, the question, one feature shown by all dicot plants. So dicotyledonous plant, you know this, broadleaf bunching.
Fibrous root.
Fibrous root is not dicot.
That's monocot. Taproot is dicot.
A vascular bundle in a shape is in a ring, right? That if you are getting that stem, if you cut it, if you're seeing that vascular bundle arranging in a ring, you can say this is dicot plant. Because when I was a student, right, we had a practical. They used to give us a piece of a stem, even though sometimes they're not showing the that green part of the stem. I can remember I got one time one small white color, just small pieces. You will not be able to understand what is this. Is it root? Is it a stem? Nothing. So what did we do when we cut the thin slices? We are carefully observing any dots. Dots are scattered or dots are in a ring. So if dots are scattered means monocot. If the dots are ring, if these dots without a microscope, you will be seeing a dot only. So under the microscope, you will be seeing there's a ring of vascular bundle. This means this is dicot plant stems. So girls, next question. During the seed development, amino acids are converted into storage protein and protease. Protease molecules become active when the seed absorbs water and at the starting time of the germination. And this figure 2.2 shows the formation of a storage protein and the protease in developing quinoa seed and the action of protease in the storage protein during germination. And if you're seeing that amino acid, you know, I told you there are 20 amino acids that which some of them are essential, some of them are non-essential. That is not the question. They're asking, these amino acids are, you know, that joining together to form a protein. Look at the protein synthesis in the seed. The part of the storage protein molecule. So this is the builder enzyme. They are working together. Okay, so these are all now. Is there anyone who is not listening? Some messages are coming. No, it's not from 10A. Okay, so this is one protein molecule. This is the part of the storage protein molecule, and this one part of a protease molecule. So both are, this is one type of enzyme. This is a protein. This is protease and this is protein. Now, both this protease are working to break down this protein into protease breakdown storage protein supplied to amino acid to the germination seed. So the amino acid in the seed, they are assembling together to form the protein molecule, storage protein molecule, and the protease enzyme also formed from the amino acid. This has been used to break down this protein, and this will supply the amino acid for the germinating seed. And Anika, you are here, Anika? Okay. So supply of amino acid in the germinating seeds, this will supply. Now, next, if you are going next phase, they're asking the question, define the term gene. What is gene, girls?
Gene is an alternate form of
a length of a DNA.
Length of DNA that codes for protein.
Yes, that codes for a protein. Now, describe the differences in the structure between the storage protein and the protease shown in figure 2.1. Look at their structure. Look at the storage protein and the protease molecule part. How can you, they're asking the differences between these two. One is a storage protein and one is protein. Now, look, by looking this two picture, can we say that storage protein and the protease has different sequences of amino acid? Right? Girls, are you getting me? Understood?
Yes, miss.
Now,
Yes, miss.
this is one point. Second point, can we say they that compose different amino acid? Okay, both have different shape and both folded differently, right? Check the diagram and tell me that you understood my point. Both have different shapes and both folded differently. Look at the arrangement. See, it is straight and it is like that. Is it clear? Hello.
Hello,
Yes,
miss.
It is, miss.
Yes, miss. It's
So
clear.
that by looking, you can understand this is that different amino acid, both their shape is different and they have folded differently. Now, state the role of mRNA and the ribosome in protein synthesis. Girls, mRNA, what they're doing, mRNA, first they will go to the nucleus, and where DNAs are there, they will be unwind, and one of the strand of the DNA will work as a template. So the mRNA will copy the best sequences which has been given by the DNA instruction, what type of protein will synthesize, and this message is mRNA copy and carries this gene from the DNA, then will come out from the nuclear through the nuclear put to the cytoplasm of the cell. Then they will go to the ribosome, they will attach to the ribosome, and they will determine the specific sequences of amino acid. I told so many things. For 1 mark, you will not write so many things. You just write mRNA carries the copy of the gene from the DNA and attached to the ribosome, where that desired just attached to the ribosome till this much is okay, because next they're asking about the ribosome. What does it do? Ribosome is a set of the protein synthesis, right? Ribosome read the messages or the codes mRNA has given, which has been brought from the DNA, right? And according to the code, that amino acids are assembled together or linked together to form the desired protein. So this thing within 2 lines you need to write for the 2 marks. No need to write so many things. For make your clear conception, I explained full. Now page number 6, if you are seeing this question, that researchers investigated the effect of pH on the activity of the protease in quinoa seed. Look at the result. Enzyme activity, and they have showed the pH. Now, could you please tell me, rather than pH, there are other factors which affect the enzyme activity? What could be?
Temperature.
Yes.
Temperature and what?
And then one more.
Air.
Air.
Concentration.
Air, moisture, and temperature.
How moisture is affecting it? It could be surface area. Think that normally surface area, right?
Without moisture, they won't even start working, right, miss?
That is not moisture without water, hydration. Okay. So you can write concentration. That is not this answer, Anika. Don't be confused, because they're asking other than pH 2 factors that affect the enzyme activity. If there is no water, the enzyme will not start working. This is not, this is after starting working, enzyme activity will affect. So surface area, type of substrate, what type of enzymes are there, volume of enzyme, concentration of enzyme, this anything you can write. And second question, describe the effect of increasing pH on the activity of protease in quinoa seed. Now here, this question is 3 marks. So, and with this data, we have to explain. Everyone look at this graph and listen. As the pH increases, the activity of the protease in quinoa seed also increases at the beginning. It is not a step increase, right? Gradually it is increasing. And what is the optimum pH, girls?
4.
Yes, 4. Excellent. So optimum pH is 4 because it gives the highest enzyme activity, and that the amount is, the measurement is 0.55 arbitrary units. From pH 4.3, look at the graph. From pH 4.3, the enzyme activity decreases, and at pH 5, the enzyme activity was 3.5 arbitrary unit. And at pH 6.5, the activity stops and it becomes 0 arbitrary unit. No activity beyond pH 6.5. So because that maybe it's too high pH for this enzyme to work. So this is the way you have to explain. I'm telling this answer one more time. As the pH increases, the activity of protease in quinoa seeds also increases at the beginning. Optimum pH is 4 because it gives the highest enzyme activity, 0.55 arbitrary unit. From pH 4.3, the enzyme activity decreases. At pH 5.0, the enzyme activity was 3.5 arbitrary unit. And at pH 6.5, the activity stops and it becomes 0 arbitrary unit. No activity beyond pH 6.5. So this is the way, by looking the graph, if you explain the whole that graph, you will get 3 out of 3. Next question, they're asking, explain the activity of protease between the pH 4 and 7. It means that, look at the graph, 4 means the optimum, right? Here they told you to explain the activity. So 4 means this is the optimum. Why the rate is high? You have to explain with the result. And at pH 7, why it has stopped? You have to explain with the result. So like more successful collision, more enzyme substrate complex, this way you have to explain this question to get 4 marks. So pH 4 is the optimum. And pH 7, the enzyme denature, right? So the active site of enzyme has changed its shape at pH 7, and enzyme substrate complex do not form, but at pH 4, enzyme will fit with the substrate and form enzyme substrate complex because the shape of active site and substrates are complementary. And the enzyme work faster because At pH 4, more effective collision between the enzyme and substrate, and none of them in that pH 7. So it won't work at pH 7, but it works highest red at pH 4. Understood? Is it clear, girls?
Yes, it is.
So I hope you
Very
understood.
clear.
Now we'll be going next question. This is phase number 8. Red blood cells in human are produced from stem cells. This stem cell, actually I told you all the cells in your body actually same set of chromosome. It has come from the zygote. Your own body has, it's a result of mitosis cell division. Meiosis only you are doing inside your body cell. It is happening only to produce egg cell, and for the man only to produce a sperm cell, haploid nucleus. So as your body cell has formed from one simple zygote, so all the body cells are same. I told you that that's the reason in inheritance chapter you learn that some cells are switching off, some gene are switching off, some are switching on. According to that switching on and switching off, they are maintaining the function, because all the cells are having same chromosome, same genetic pattern, but some of the places the responsible gene was switched on, that function carried out, and others are switched off. You know this thing. The melanin, the gene which is responsible for digestion of or secretion of the pepsin, that is also that present in your skin cell, but it is not working over there. It was switched off. So stem cell, the first beginning, the cell in the zygote, in the embryo, that is the stem cell. Before specialization, before differentiation, all the cells were same. But this cell we call embryonic stem cell. So embryo, all the cells are same. From the embryo, some of the cell turn into bone tissue, some of the cell turn into heart tissue, some of the tissue turn muscle tissue, nerve tissue. All the tissues, all the cells has been formed from the stem cell only. But according to the function, the feature, the structure of the cells has difference that occurs also. Now the question is, girls, as all the cells in your body has come from the same the source of cell, right, from the zygote, so all the cells, their basic configuration actually same, but according to the function, look, the skin cell has one shape, nerve cell has the fibery shape, so muscle tissue have one type of shape. So once the stem cell has different shape, it will not change. So if the cell turn into a blood cell, the blood cell will not change. So now what happened, nowadays the scientists are doing stem cell research. People are doing the research and they are trying to do stem cell therapy to make the organ work, especially the inactive organ, because stem cell has regenerate capacity, but still is in research condition, and many people ethically they don't support it, because using stem cell means you are killing potential human being, because we all started from an embryo, right? If you are killing embryo means you are killing a human being. That ethically none of the religion has support this. Now in this diagram, if you are looking, before this is explaining this diagram, I want to tell you the question. Use this information and describe how red blood cells are produced and explain how they are adapted to their function. This question has 2 parts and total mark is 6. First part, they're asking how red blood cells are produced, and second part, they're asking explain how they are adapted to their function. So look, first I will explain how the red blood cells are produced. Look, the production. See, the stem cells are, girls, could you please tell me how see, this is the one stem cell and becoming many stem cells. What is the name of this type of cell division?
Mitosis.
Excellent, excellent. Stem cell divide by mitosis division and form genetically identical cells. Look, there are many genetically identical cells. One of the cell, look at the arrow, one of the cell specialized to produce red blood cell and others continuing as a stem cell. So first what happened, look, the nucleus bursts off because mature red blood cells do not have nucleus. After removing the nucleus, mitochondria has been broken down and digested because there is no mitochondria in mature red blood cell. Now red blood cell has formed. So in our question, first part we have completed. Look, how red blood cells are produced. We have explained. Stem cell has divided by mitosis division and formed genetically identical cells. Only one of the stem cell has specialized to produce red blood cells and other continue to be stem cells. First, the nucleus of the stem cell bursts off because there is no nucleus in mature red blood cell. After removing the nucleus, that mitochondria has broken down and digested because there is no mitochondria in the mature red blood cell. Now they're asking the adaptation of the red blood cell with the function. This you did that when you are solving chapter 2, the adaptation of different specialized cell. So from there you can write, see the loss of nucleus and mitochondria give space for more hemoglobin to transport more oxygen for the red blood cell, and also by concave shape of red blood cell also increase or gives them more surface area or large surface area compared to the volume ratio. This shape also helps them to pass through the capillary and for the diffusion of oxygen. Is it clear, girls? Understood?
Yes, miss. Yes, miss.
Now
Miss,
that, yes, miss.
the
Yes.
enzyme question.
Yes, sure.
Miss, do you mind
No.
explaining it again?
Okay, no problem. That let me finish this question. Okay, so this is clear, right? I'll come back. I'll go back to the exam question again. No problem. So this is clear, girls. This is the one of the big question about the stem cell. After that, I have seen few stem cell questions are coming, but that is small. Describe how red blood cells are produced. You understood, and their adaptation. Adaptation is from your notes, but how red blood cells are produced, you have to explain from the diagram. Now name the liquid part of the blood, that is plasma, you know. Explain why stem cells are found in the skin, because you are losing the epidermal layer of the skin every time. So stem cells are found under the epidermal layer of the skin to replace the cells that die, rubbed off or exfoliated. It also works to heal your wound on the skin. So girls, this 2 marks, 2-3 points are enough to get. I told you many things, right? So I will go back to the exam question that that you need. Did I explain this one or from the beginning?
No, miss, this one.
This one,
question.
okay. So this one, the question, do you need this answer? Shall I tell it a bit slow so that if you you can write the important points only, right? No need to write the full answer, this point. Look, activity 4 and 7. So 4 is optimum. So you will explain what happened in the optimum temperature that increase the enzyme function. And 7 means denature. So what happened denaturation time so that enzyme stop working? This 2 part of this question. Let's see. Let's see the diagram. So you will write pH 4 is optimum pH and pH 7 the enzyme denature, and the active side of the enzyme has changed its shape due to this high pH, and enzyme substrate complex are not forming. So that at 7 enzymes are not at pH 7, it is not working, and there is no successful collision between enzyme and substrate. 7 has done. Now pH 4. pH 4 is the optimum because that enzyme fit with the substrate and form enzyme substrate complex because the shape, both the shape, the active side and the substrate shapes are complementary, and there is more effective collision between enzyme and substrate, which makes the enzyme work faster and gives the product more quickly. So as enzyme work faster in this pH 4, this is optimum pH. Clear? Hello?
Yes, miss.
Understood?
Yes,
Shall I? Okay. If you have any question that I have solved till page 9, if you have any question, you can ask me. No problem. Don't hide your problem. Asking question is not a problem. If you don't understand, this is your right to ask. But if you don't understand and then keeping quiet, you are increasing your problem. Now, state the nutrient that is the deficient in the diet when anemia occur. What is
Iron.
the iron? Excellent, iron. And what is the symptom?
Shortness of breath.
Tired.
Yes,
Fatigue.
continuous fatigue, right? Yeah. Why you will be tired? You will feel always tired if you're anemic. Could you please explain?
Because hemoglobin.
Because your blood cannot carry much oxygen, and then you whatever Nizam is saying.
Less oxygen.
Enough
what
oxygen is
does
not
it do?
transported.
If your blood is transporting less oxygen, then why the fatigue will come? Why you will be weak?
Because you don't. Because we
Because
need oxygen
enough
for breaking up the
glucose is not
energy.
broken down to
For breaking up the energy,
Less
and your
respiration.
glucose won't be broken
Excellent.
down for.
Someone is messaging me. Okay, no problem. So excellent, girls. You are right. So you will be tired. You will feel tired every time. You will be weak every time because the sufficient amount of energy you are not getting, because respiration need oxygen, right? So as the lack of oxygen, how you are going to get the energy? Good. Now, some people have sickle cell anemia. Discuss the cause of this type of anemia. See, girls, suppose in your whole family, there is no history of having sickle cell anemia. But when suppose one couple, imagine one couple, this couple in maternal side, paternal side, none of them have the history of having this sickle cell anemic patient, but this couple got one child with this symptom, and this is very dangerous, and the child is very sick. Why? And how does it happen? None of the family are having this problem, but this couple, think about the family tree. What could be the reason so that this couple will get 25% child with the sickle cell anemia? Because both the parents are heterozygous. So mom and dad both are having these characteristics, but it was recessive. It is codominant, right? So when the persons was heterozygous, they have this sickle cell anemic trait, but it was not creating problem in their life cycle because they have enough normal red blood cell which could support their life. So they will not be able to understand they are carrier. So when two carrier people, I told you this is the genetic that inheritance rules, right? When two heterozygous peoples are that married, and if they going to that produce a child, 25% child will be recessive characteristics. So two heterozygous organism cross, it will give you 3:1 ratio. That one means 25%. That will come to the recessive characteristics. So if the family do not contain this symptom also, or none of the member is having this sickle cell anemia problem, 25% child will get this. And another thing is 25% child, those who are having sickle cell anemia, they will not survive for a long time. Most of the time, the sickle cell anemic patient die before puberty. So there is no chance to go to the next generation through this sickle cell anemic patient. Only it will go through the heterozygous people. So describe the cause of this kind of anemia. This is not cause, this cause is mutation, right? Why that I was explaining, if the family members are not having, even though you are getting, but describe the cause. What is the cause of anemia? This is mutation. Due to the mutation, which changes the DNA base sequences, and hemoglobin become mutate and become HBS. So RBC become distorted and sickle shape. RBC cannot supply sufficient oxygen to the body cell and can block the capillaries. The person, those who receive this HBS, HBS allele from the parents, they will not survive. Even the mutation, if it is occur like that during the pregnancy time, if the mutation occurs, okay, that is called gene mutation, right? Due to the gene mutation, if the child is getting this thing, that this child will die before puberty. So that this prevent their normal lifespan, and this mutant allele couldn't pass to their child as they passed away before the puberty. People can inherit this disease also through the heterozygous parent. So mutation, if the mutation occurs, you will get this, or you can inherit from the heterozygous parent. This is the cause of this kind of sickle cell anemia. I hope you understood. So our question number 10 has over. We'll be going to next question, page number 11. Look at this photograph. This is yellow-shouldered Amazon, Amazona barbadensis. This is the scientific name of this bird, a species of parrot found along the Venezuela coast of the Caribbean. Girls, I have short of time, less than 1 minute. I'm coming with the new link, okay? Just a minute. I'll start from page number 4, that question number 4.
Okay.
Good afternoon, girls. Our second session. I was stopped from question number 4. Look at this question. This is the photograph of a yellow-shouldered Amazon. Scientific name is Amazona barbadensis. This is the species of parrot found in Venezuela coast of the Caribbean area. Now, this picture, which vertebrate group, sir, is it? Could you please tell me?
Bird.
Bird or avis, right? And you need to tell 2 features that are used to classify this organism into the group of birds.
Wings. Feathers.
Wings, that wing that I prefer not to give you here, because feathers, big bill, because insects are also having wings, right? So the questions, they are used to classify animals, makes it separate. So a special feature, this are only you will get to the that birds only. You can write they do not have teeth, they have feathers, and they have beak or they have bill. Which organism? This organism have beak, right? This have beak, and some of the birds like duck, they have bill also. So you understood this question. Now next page, this page, the second question, if we are talking, see, this species is subdivided into several populations on the mainland and on the island of Margarita and Bonaire. This is the 2 places of the island. Scientists believe that yellow-shouldered Amazons rarely travel between these places. This is not a question, girls. This is the sentences they're using to make you confused. So don't confuse with these sentences. Your question is, explain what biologists mean when they refer to population of animals such as that Amazona barbadensis. It means you will give the definition of the population and you will give example of this yellow-shouldered bird with an example of the population, like that. Let me tell you this answer. Biologists mean group of individual animals when a group of same species living in the same habitat at the same time is called population. Like all the Amazona barbadensis living in one area is called their population. Understood? So if instead of this bird, another example could there, you will answer like that. There are many questions that are coming like that. You need to explain with the definition and you will add this as an example with the definition. So is it clear? Let's go to the next page, page number 12. The number of yellow-shouldered Amazons on Margarita Island had decreased to 700 parrots by 1989. The population then increased to 1,600 parrots by 2009. So it means maybe they have done the captive breeding program, right? Now, suggest why the part of this increase was due to the release of captive bred parrots on the island. This is one of the few successful release programs of parrot. A similar release program in Arizona in 1918th century of the different species of the parrot was not successful. Now the question is, suggest why release programs of captive bred parrots were not successful. Thus, in the notes, you learn about the disadvantages, right? So you have to, this thing, maximum, this answer, you have to understand and you have to explain. Look, one area it is successful, the captive breeding program. Another area it is not successful. Why? Minimum 2 points we have to give. So first point we can give like that. The parrot may killed by predators. They are not able to find food or more prone to diseases, right? Actually, I gave 2 points. And you know that within the second point, you can write that competition with the new species and that captive animals are unable to adjust or to cope in wild and not integrated with the wild population of the parrot. They couldn't adjust because while you are keeping this organism in the captive breeding program, you are giving them whatever they need, their food, their shelter, everything you are providing artificially. But when they will be in the natural ecosystem, they have to obtain their food by themselves, by fighting with the existing animals. So if you are releasing an animal from the captive breeding to the wild, sometimes they couldn't adjust because they don't know how to fight with the other organism for their food, for their shelter, for everything. And captive breeding, another disadvantage is you are keeping less number of organisms. So when the reproduction occurs with less number of organisms, it created genetic variation. So less genetic variation. So less genetic variation and also increases the chance of genetic disorder. See what happened, you know, this captive breeding program example, if I'm telling you, I told you sometime I used to distribute food outside to the cat, right? So my cat also now is outside. I don't know where he is because of this virus fear. Nobody's agreeing. So I'm giving food outside. But what happened, you know, I have seen that when I have given the food, the street cat, immediately they're coming and they are fighting with each other. And some of the cats are sitting, they are not having the guts to take the food because one very powerful and energetic cat is taking the food. So you, after taking, if the leftovers are there, then that other cat will get the chance to eat. So this is like that. You have to fight with food, for the food, for shelter, for everything with the existing animal. Otherwise, you cannot adjust. So this is the reason that release program for captive bred parrot were not successful in one area and another area was successful. Now, the second question they're asking that some captive breeding program involved very small number of animals. Outline the disadvantages of this type of program. So captive breeding disadvantages I gave it in the notes. You just need to write 3 points like there is not enough animal to breed in captive breeding program. And due to inbreeding or breed between a small number of the animals causes less genetic variation and increases the risk of genetic diseases or genetic disorder. And animal less likely to adapt or cope with the changes in the environment like habitat change and habitat change or cannot adjust with the introduction of the new species. It can happen, right? Migration of the organisms are going on every time. If the new species comes to the environment, they couldn't adjust with them also. And third point, this program is very costly. Sometimes personally, nobody can support this captive breeding program. They need government support. Sometimes government also couldn't support in large scale. So these are the 3 disadvantages. One, genetic disorder, one, adjustment with the environment, and third is the costly. So these 3 points, if you're explaining here, you will get 3 out of 3. Now, this says many biologists think that it is better to conserve ecosystem rather than individual species, such as parrot. And that's true. If you are conserving ecosystem, rather than conserving only one species like parrot, it will be more advantageous. If you are conserving ecosystem means all the biodiversity you are helping to maintain and all the biodiversity supported, many organisms, even the cycle, these all you are So question is, explain the advantages of conserving ecosystem. You have these notes also. Or do you have or you forgot? Girls, don't tell that I don't have these notes, please. So
No, we have.
Yes, you have this. So for 3 points, I'll tell you, conserving ecosystem, maintain biodiversity and saves many species and prevent extinction. 1 point. It prevents disruption of food chain and food web and gives shelter and breeding ground for many species. 2 points. Conserving ecosystem also provides services for humans like reduce natural disaster, greenhouse effect, maintain the balance of different nutrient cycle like carbon cycle, water cycle, provide food, fuel, drugs, and raw materials. 3 points. You will get full 3 marks. And if you want to write, it can also use for future genetic engineering, and it can also use for ecotourism or recreation. These also you can write. So 3 points for advantages of conserving ecosystem will give you 3 marks. Now I will be moving on next page, page number 14. An investigation stated that the effect of fertilizers on grass yield and species diversity in a grassland ecosystem. Some plots within the grassland were treated with fertilizers containing nitrogen, magnesium, phosphate, and other control plot. It means they have actually used 2 plots here. One was with fertilizer and one was without fertilizer. So without fertilizer, you can say this is a control plot. And the average that yield was 1,733, the plots with fertilizer, and the plots without fertilizer, the control plots, 1,009. The unit they had given that GM -2, ER -1. You can check the data. Now, what is the question? Calculate the difference between the average yield of the 2 plots within the field as a percentage of average yield of the control plot. Show your working and give the answer to the nearest whole number. So how can you do this thing if I'm showing you this? Now I will show you in the whiteboard this calculation. Let's
72%.
Yeah,
72.
what 72 did you do, or you were seeing the mark scheme?
No, I have done.
You have done, good. So let us do also. So 1733 minus 1009. The result is 724. You have to divide with 1009, and you have to, the result, whatever the result, you have to multiply
with 100.
With 100. Then let me use the calculator. 724 divided by 1000.
Miss, it's 71.75, which is 70%.
It will be 0.71754. And now if you are multiplying with 100, your result will be 71.754. And you need to use whole number 72. This is the answer. So this is actually the formula you learned, that original value, final value minus original value divided by original value multiply 100. That is actually the answer. This same way we will be doing it. Now let us coming back to the question again, the one we are solving. So you got this answer, that question number 5A. Now next question, if we are going down, let's see the next question. They're asking, explain why average yield increased as a result of adding fertilizer. See, fertilizer means you are giving the nutrients the plant need to grow, right? So of course they will get the nutrients, they will grow faster, their photosynthesis will be faster, they will give you more product. And if you're not using fertilizer, they will not get the essential nutrients which help them to grow faster. So for the 3 marks, how you are going to write? Fertilizer provides different ions. Salts and minerals are required by the plants, like the plant need nitrate ions to make amino acid, protein, DNA, RNA. They use this protein for their growth. Magnesium is needed for making chlorophyll.
Chlorophyll.
Yeah, increases the rate of photosynthesis. And this all will increase the average amount of yield. So adding fertilizer means they are giving off lots of key nutrients to the plant. Like the fertilizer provide nitrate ions, which plant use to make protein, amino acid, protein, DNA, RNA, which is needed, protein is needed for their growth. And magnesium also, that fertilizer provide magnesium, which is used, the plant used to make chlorophyll, more chlorophyll, more photosynthesis, and the more average, more crop will produce, right? So the average amount of yield will increase. So you have to explain in this way. Now, state the name of the process that occurs when fertilizers washed off into the land and rivers, which causes an algal bloom. Girls, what is it? Eutrophication. Excellent. Now, if you are looking the next question, this is also simple, but it's looking, oh my God, what is this question? But see, the researchers counted the number of different plant species in each of the plot in the grassland to determine the species diversity. Counting starting at the beginning of the growing season was repeated every 3 weeks. They have counted actually the average number of plant species in each type of plot shown in figure 5.1. Look, the plots without fertilizer, the plots with fertilizer. The black color, and if they have told you to just to describe the effect of fertilizer. They're not asking you that with the reason. So look, with fertilizer, it is increasing, then decreasing, again increasing, decreasing. And plot with fertilizer, increasing, decreasing, and the last one, a little bit increase. So whatever the pattern you are seeing, you need to explain with the data. Like fertilizer decreases species diversity. As time increases, the diversity increases. From 6 weeks, look at the graph. From 6 weeks, diversity decreases. At 21 weeks, the differences is greater. The average number of plant species for plots without fertilizer was 15, and the plot with fertilizer was 3. Just give unit that meter per one. Give the unit like that. Just a minute. Just a minute. Now see, girls, that again I'm telling you this answer. Fertilizer decreases the species diversity. As time increases, the diversity increases. From 6 weeks, diversity decreases. At 21 weeks, the differences is greater, and the average number of plant species of the plot without fertilizer was 15. And plots with fertilizer was 3. Add unit also. At 21 weeks, the plot without fertilizer, the species diversity increases, and at 24 weeks, it decreases. But with fertilizer, is not following the same trend. So whatever you are seeing in the graph, 1 or 2 data you will give and just explain increase, decrease, increase, decrease. That's it. Okay, understood? Is it clear? Next question,
yes.
I will start from page 16 tomorrow. Tomorrow also you have class. Okay, tomorrow I'll solve on paper 6. Then I will, first I will start paper 6, then I will be with this. Second session, I will come with this question. Okay, from page number 16, 17, and then 18 I will explain tomorrow. So I'll give you the link for paper 6, one variant, then I will solve this. Okay, girls? So please, we all are instructed to request you all to clear your dues. Please clear your tuition fees. The school need to run without any that problem. So all the girls, if you are not paying after 10th April, they will not allow you to enter any virtual classes. Thank you, girls, and goodbye.
Thank you, Miss. Goodbye.
See you tomorrow.
Thank you, Miss.
Thank you. And girls, I
Yes.
can understand many girls are not that coming, because 13 students, right? Repeatedly they are not coming. I need to point out the girls, those who are not coming regularly. Some are coming and going. I can understand the reason. The regularly few girls are not attending the class. Sarah Azam Khan.
I am a good girl.
I have never seen Sarah is in my class.
I'm very good girl.
Yeah, you are a really good girl. Could you please inform Sarah Azam Khan? I have never seen that this is the fourth week maybe I'm doing this online classes, but I didn't see her. So inform her to inform us why, because as a homeroom teacher also I have to inform. Sarah Azam Khan, regularly she is absent. She is absent in other classes also.
Yes.
Then if anybody knows her, please give a message personally. Huh? Okay, thank you, girls. Bye.
Thank you, Miss. Goodbye.
Oh, who's this?